Welcome back. Today we are going to be talking about transport. We are going to be reviewing some of the key ideas we had from this unit. First off, what is the difference between facilitated diffusion and regular diffusion? With facilitated diffusion, large stuff, so this is going to be like the starch molecules that we had in the plasmolysis lab, where basically you had the dialysis tubing with the starch inside and the iodine outside. Now, the starch was too large, so it couldn't leave the membrane. Now, in real life, if the starch wanted to get out of the cell, it would need to be brought in and out of the cell through the cell membrane with the carrier protein. Basically, these things are kind of in the middle of the cell. They kind of look like pores with the lipid bilayer. Certain big items need to pass through those. The easiest way I can put it is if you're playing the game Mario. Mario couldn't fit through certain things, but he could be transported in and out of really cool worlds by going through those green tubes. So he could go from the sky world to land, back down to a dungeon, and back and forth by those little tubes. So the starch, to get back in and out of a cell, would need to be carried through that by a carrier protein. So that is going to be these little things right here. Moving on, regular diffusion. This is basically going to be small stuff that can pass freely through the cell membrane with no problem. So an example would be that with the plasmolysis lab would be the iodine being able to sneak through the cell membrane, which was the dialysis tubing, and be able to get in and out freely with no problem. Now, basically, remember that it's going to be going from an area of high concentration to an area of low concentration. So the area of high concentration in our lab was basically on the outside where all the iodine was, and it wanted to go down the hill, so it went into the membrane. So that was inside of the dialysis tubing. Moving on, what is the main difference between diffusion and osmosis? Really, they are pretty much the same thing. Diffusion is when stuff goes back and forth in and out of the cell with no problem. So that's basically anything that's small enough to go in back and forth freely that the cell membrane will allow, it will happen. So that's diffusion. Osmosis is basically going to be the same thing, but if you look, this is when water goes back and forth in and out of the cell with no problem. So diffusion, anything else. Osmosis, simply water. And remember, that's going to be going from high concentration to low concentration. What is bulk transport? Now, easiest way to think of bulk is when you want to buy in bulk. So like if you were going to a wholesales club, instead of buying like 1 pound of cheese and 1 pound of meat and you're trying to make tacos, you decide to buy a whole bunch of meat and a whole bunch of cheese. Or in a cell's case, you basically need proteins and sugars in order to survive. So you decide to go to Costco and buy 50 pounds of sugar and a whole cow. You are buying in bulk. Now, what's the problem when you buy in bulk? Again, when you buy in bulk, you need to get it to somewhere, and you need to get there fast. Again, moving in and out of the cell in bulk is going to require some special processes. And that's what we're going to talk about our next thing. So, bulk, when you buy in bulk or when you're trying to get bulk stuff inside and outside of the cell, is going to call for two special types of transport. First one will be endocytosis. Endocytosis is the movement of stuff into a cell membrane via member vesicles. So basically, looking at these things, the big bulky items such as the protein or sugar comes around, and the membrane basically makes a little thing around it, kind of like packs it in a baggie, and then it goes into the cell. Kind of quickly see an animation, just to keep it simple. Stuff comes in, gets packaged, gets sent into the cell. That's called endocytosis. The opposite is called exocytosis, so exiting. So exit, or leaving the cell. Exocytosis is the movement of stuff out of the cell via the cell membrane. Again, I'm going to go through the big item here, which might be a protein of some type, or sugar, is basically wrapped and packaged and sent out of its way through the cell. Special thing, you can watch a video on it if you want. Just to keep it simple here, we are just basically saying it's being wrapped like in a bag. Finally, different types of endocytosis. There's phagocytosis, meaning the cell is eating. So basically when the cell is bringing in food that it needs to consume, that would be called phagocytosis. When the cell is drinking, that will be called phagocytosis, meaning the cell is drinking stuff. So basically you can use different types of things the cell is trying to survive. If you have any questions, please ask. Hope this has been helpful with your review.